All right. So a lot of, I don't know if you printed out my lecture notes the first time I posted them, a lot of these slides probably aren't in there. A lot of them are, and I actually just added a bunch more today. So I will be uploading a version 3 of Chapter 14 for this. So don't freak out if a bunch of these are things you haven't seen, but there's going to be some new stuff in here. And the first half of what I'm going to talk about today is in Chapter 4. It's talking a bit about systematics, taxonomy, hierarchical naming, classification. And then the second half of today's lecture, I'm going to be talking kind of about biodiversity in general and about species in general. Because what we're going to be going into in the next few chapters, so chapter 15, 16, and let me see where else. So like 17 as well. Let's see, 15, 16, and 17 is we're going to be looking at different groups of organisms. So we're going to start off on this Thursday talking about bacteria, then we'll look at algae, we'll look at plants and fungi, and then we'll look at animals kind of after the next test. I'm going to split it up a bit in that case. So before we get into learning kind of specifics about anything, and we're really not going to be talking about specifics about any of these groups, we're going to learn general kind of themes about all these groups and a little bit about the biodiversity of life on Earth. Before we get into that, I want to spend today talking about the science of classification, which is what systematics is. So systematics is the science of classifying organisms. And we as humans love to do this. This is really something that as humans we are kind of programmed in some sense to do, is to take things and to group them up and to group them by similarities. So we love to take even things that have no similarities at all and say, this is how I'm going to group these together. If any of you are real neat freaks, this is probably what you love to do at your house. You like to put all the pencils in one place, all the cups in another place, all the condiments in one place in the fridge or something. We like to classify and group things according to their similarities. In biology, the study of, or the systematics is more than just classifying organisms based on any random similarities, because as we're going to see, we can group things based on lots of different characteristics. It's actually grouping things based on evolutionary relationships. So we're going to learn a little bit about how things are classified and how this classification system relates to everything we've already talked about in Chapter 13 and so far in Chapter 14, which is evolution of organisms. So systematics, a big part of that is taxonomy. And taxonomy is really the science of identifying and naming things and putting them in a category. So systematics includes taxonomy, and it's kind of taxonomy linked with evolutionary relationships. So a lot of what we're going to be going into in the next few chapters is basically taxonomy. I'm going to tell you how we identify something as an algae, how we identify something as an animal, how we identify something as a plant, and the different groups of plants and what makes them different. And I'm going to tell you some of the names of these different organisms and what makes them different. And a lot of that is taxonomy. In biology, we seek to tie taxonomy to systematics, to some kind of evolutionary context. And that's kind of what I'm going to be talking about a little bit today. So ultimately, with biological organisms, the goal of a taxonomist is to put a name on something. And a name in taxonomy has a lot of meaning. So you've probably heard of scientific names for things. So humans have a scientific name, Homo sapiens, right? So lots of birds you might know them by something called the common name, but they also have a scientific name. A scientific name is designed to ease communication by unambiguously identifying organisms. So there are cases, and there's a lot of cases in kind of commonly identified things like birds and plants and things like that that you run into in your everyday life, where there'll be multiple things with the same name from different cultures. Different people in different parts of the world will have the same name for different things. A scientific name should be unambiguous, so it should be one single name for one single species, and that's the only name we ever use for that species scientifically. And the goal is to make it easier to recognize, well, one, the discovery of a new species, and also easier to recognize that you're communicating about the same thing with someone else. So if I wanted to talk to someone in Australia about a particular species and work that I've been doing on that species, by using the scientific name, I can be sure that they know what I'm talking about. If I just say the large furry mammal, that could be anything. But if I say, certain species of Ursus, then they know exactly what I was saying. So where these scientific names come from is something we've already mentioned before in this class. Comes back to Carolus Linnaeus in the 1700s, where he proposed a taxonomic system based on a two-part naming system for each species and a hierarchical classification of species into broader groups of organisms. So that's where Homo sapiens come from. That's the binomial, the two-part name. And we're going to see in the next few slides where this comes from, that there is a hierarchical naming system. Now, Linnaeus never intended for this to be based on evolution at all, but we have modified his system of naming things from what he proposed in the 1700s to what we currently use today, so that it does represent evolution. So there is an evolutionary context with which we put these naming system is. So Linnaeus used a two-part name system, so each species has two names that it's known by, a genus and a species. Here we go, here's what it does. So each species is assigned a two-part kind of Latinized name or binomial consisting of a genus and a species name. So this, for example, the scientific name for humans is Homo sapiens. A lot of this kind of specifics that are on some of these slides that I grabbed are not that particular, but you will notice that a lot of these are Latinized, so they're not necessarily Latin words, but they're words that have been written in a Latin style. So when we come up with new names for new species, we always have to add a Latin kind of ending to it. It just makes things all uniform amongst all scientists. You'll usually see scientific names italicized, given a Latin ending, and you'll notice that the first letter of the genus is capitalized and the species name is always in lowercase. And these are just some common things about the way that we write scientific names to make them consistent between different groups that are talking about them. Okay, so every species has a species name and a genus name, and then some other parts I'm going to talk about in a few more slides. They all have a binomial. How do we use this scientific naming system to tell us about the relatedness of things? Well, organisms that are closely related will share similarities in their scientific names. So this genus name, Homo, that we have, so we're Homo sapiens, Homo is our genus. There are other organisms that were in the same genus as us. There's no currently living species that has that same genus, but there are things like Homo erectus, Homo habilis. Those are other human-like species that share our genus names that were closely related to us. So species that are closely related are placed in to the same genus. So they'll have different species names, but the same genus name. So I have an example with some pictures in the next slide of different cat species and their species, genus and species names. So here are 4 different species: leopards, tigers, lions, and jaguars. And you'll notice that leopards their scientific name is Panthera pardus, lions are Panthera leo, jaguars are Panthera onca, and tigers are Panthera tigris. So these all have the same genus, but they have different species names. And so if I'm communicating with someone about Panthera onca, they know what I'm talking about. There's actually a lot of different things that have the name jaguar, or there's different names for jaguars out there from different parts of the world. But Panthera onca is the scientific name for this particular species. And since all of these have the same genus, the same Panthera parts of their name, it indicates that they're all closely related, that they share common ancestors, that they're pretty closely related. So it doesn't just stop with genus. There's actually quite a hierarchical classification system that we use that includes these other terms as well, which you've probably heard of before, that are broader than genus. So families. So might be several genera that are part of the same family, and they'd all be closely related to each other. Yeah.
So like on the last slide, the tiger one. For different kinds of tigers, they all still the same, like whatever.
If they're the same species. So there are different species of tiger.
So that was just like the basic.
That was just one tiger.
Tigers.
Yeah.
Like, I don't know what you call it, breeds of tigers. I guess.
Yeah. So well, there's a difference there. So breeds. So like dog breeds.
Or like,
There's.
I just watched a show of
Yeah.
tigers. How there's like 9
Okay.
different tigers. I mean, like
Yeah.
all Panthera, whatever tiger is, so they have a different one for each one.
Yeah, that's a really good question. I don't know that. I don't know much about tigers specifically, so I don't know how different they are. Yeah.
Like, are they all the same? Like, or do they have like the tigress at the end? Does that change into something different?
Well, if they were not interbreeding and they were separate species, then there would be different species names, and that's a case. We should look that up to find out if that is. I'll have to look that up. I'll have to keep that in mind, and definitely next class I'll have an answer for you for that. But that happens a lot, is that there'll be all these different things that we just say, oh, that's a tiger, and we don't know, well, is that all the same species or are they separate species? So if they're all interbreeding and they all share genetics and they're all similar, they're just different varieties within a species, then they'd all have the same species name. But if they are separate, they don't interbreed, there's barriers to them mixing genetics, they just happen to look similar enough that everybody uses the same common name, they might have different species names, in which case they would be separate species. And so that's a case where it's confusing. Yeah, it's confusing. I don't know. I'd like to actually look that up because I'm not an animal guy, so I don't know as much as I probably should about that kind of thing. But we can definitely find that out. So yes, and that's something I'm going to talk about later on in this talk. I'm actually going to look at my field, and it's very, very hard to tell things apart, which is actually why in my field in seaweed, we almost never use common names because common names are so confusing that it's hard to tell, well, are you talking about 2 different things or the same thing? And you probably know that here, like we have different names for the same things. So there's certain species that will be called different things in different parts of the states even. We'll have some examples as we go along. Okay, so things that are in the same genus are very closely related. Closely related genera will be grouped together into families, closely related families into orders, and closely related orders into classes, closely related classes into phylums, and so on and so forth. So every time you move up an order, or you move up from genus to family to order to class to phylum to kingdom, you're getting broader and broader and broader. So here's kind of an example of this. So this common leopard, Panthera pardus, is a particular species. It's in the genus Panthera, so there are other cats that are very closely related to it that are in the genus Panthera. There are also cats, things that we would call cats, that are further away from this. So they share a lot of similarities with things like tigers and lions, but they're different enough that they've been put into a different family. So we have Felidae, which are just is the family, whereas Panthera is only one genus within Felidae. There are also other carnivores besides cats. So there are dogs. There are other carnivorous kind of animals that share a lot of similarities with cats, but are not cats. And these would be in the order Carnivora. And then there are other mammals. So deer and cows and humans share some similarities with the carnivores, but we're not the same. So they're in the class Mammalia. Then there are other organisms that are not mammals, but have spinal cords, like fish, that share some similarities with mammals, but are different. And we're going to learn more about this when we get to the animal stage. And they would be all in the phylum Chordata. And then there are other animals that don't have spinal cords, that they are invertebrate animals. So you may know some of these things. So some of the insects and sea stars and lobsters and things like that, that are animals, but they're not chordates. They're going to be in the kingdom Animalia. And then there are other eukaryotes that are not animals. So plants, a lot of plants, so some algae, things like that, will be in the domain Eukarya. So every time you go up, or you go kind of down on this graphic, you're getting broader and broader and broader. Every time you go up on this graphic, you are getting more and more specialized and more closely related to the eventual species. So we can put things into groups. And this would be similar to saying you're a student in this class versus you're a student that's taking BSC 1005 versus you're a student at Florida Southwestern versus you're a student at a school in Florida versus you're a student in North America or something like that. So you're getting broader and broader and including more and more people in that group. It's the same thing here. As you go from species to genus to family to order to class to phylum to kingdom, you're getting broader and broader and including more distant related individuals. Okay, so does this make some sense? What we're going to be doing over the next few chapters is actually looking at several different, first we're going to start out with the domain bacteria, so a different domain than eukarya, and then we're going to explore some of the kingdoms in the domain eukarya. We're going to look at plantae, we're going to look at fungi, and we're going to look at animalia, and then we're going to look at several different types of animals. So we're going to be actually looking at several of the broad groups, and in each of those lectures that I do on each of those chapters, we're going to explore some of the diversity within that. But there's an incredible amount of diversity out on Earth, so there's really no way we'd even scratch the surface of it. But I will give you an idea of all the different types of organisms that are out there and some of the higher level classifications that we'll be looking at. Okay. So we're going to delve a little bit deeper into systematics, which is using taxonomy, which is what we just kind of talked about, which is the naming of things and the grouping of things, and organizing and interpreting biological diversity based on an evolutionary context of it. So systematics is the science dedicated to discovering, organizing, and interpreting biological diversity, and it's often in an evolutionary context, you might say. Yeah, I've already had this before, but I might have written it slightly different in the 2 slides, so don't worry about it. There's many different kinds of it. Okay, so if we zoom way, way out, this is what biological diversity looks like. There is a theoretical common ancestor for all life that we really have no idea what it looks like. It's very hard to describe the ancestor of all life. based on current living species. There are three major domains. There are bacteria, archaea, and eukarya. And over the next few chapters, we're going to start off looking at bacteria and some of the diversity in bacteria. We're going to look at some of the highlighted branches in eukarya, so land plants, animals, fungi, and we're going to kind of group all the rest of them together in the algae, although it includes a whole bunch of diversity that it's going to be very hard to talk about. So what you're looking at right now is a biological tree, or an evolutionary tree. And what evolutionary trees do, and I want to talk a little bit about these, and I'm going to use a kind of simplified version here to talk about it. And we've talked briefly about this, but I want to get into more detail because you'll be seeing these as we go through the next chapter. An evolutionary tree shows relatedness of currently living species, so based on their evolutionary past. So everything on the very right side of these trees are present species. So here I just have these theoretical colored animals. All these different colored animals are the living species. To the left, as you go left on a tree, you're going further and further into the past. And if you go far enough into the past, you'll find a common ancestor that was a common ancestor for all of the species. So on the right are what are called the descendant species, and on the left would be an indication of the common ancestors. So this is also called the root of the tree, and these would be the tips. Each of these little connections here, like this, are called nodes, and those represent a speciation event. And this is something we've talked about already in this chapter. We looked at when those two bird populations started to diverge. We would consider that a potential speciation event. So that's an event that separates populations into two separate species. So that was the last time that those two species shared a common ancestor. So this blue line represents the most recent common ancestor for both the green and the black animal there. Organisms that are closely related or next to each other on a tree and share a common ancestor that's more recent than they share with anybody else, we call those sister species all the time. So this is a common term used for closely related species, is sister species. So this is a speciation event as the nodes, and before the node, they shared a common ancestor. So if we think about the past for these two species, the dotted line here represents shared history. So the dashed line. So both species were the same. It was one species, basically, during the time where that dashed line existed. They shared a common evolutionary past at that time, a common history. At that speciation point, that's where they separated, and the green line represents the unique history of the green species, and the black line represents the unique history of the black species. Hopefully this is pretty clear from this, right? Okay. So this side over here, they had common ancestors. Okay. So some other terminology that you'll hear, and you'll hear me use sometimes. A group that includes the common ancestor of all the species that you are looking at is called a clade. So a clade is a grouping that includes a common ancestor as well as all the descendants, all the living descendants of that common ancestor. So this would be a clade here. All of these tips can be traced back to the same common ancestor. Okay. Clades are something we call them monophyletic. And don't I'm not going to test you on a lot of these terms because they get really complicated. But monophyletic means that it has this clade all shares the same evolutionary past prior to that common ancestor. So they're monophyletic, they're one clade. So here's some examples of clades. So the blue box represents a clade. The brown box represents a clade. Even the pink box represents a clade because the organism at that node, they all have a common ancestor. Yes, it's only one species, but yes, they could be considered a clade as well. And just like you could consider the blue and the brown one a clade. However, this would not be considered a clade. So this pink box, because it does not include the common ancestor for these species. So if I wanted to include all three of these species that I've highlighted as a clade, how far back would I have to go to make them part of the same clade?
Just the first two?
Well, my question is, these three, so I've highlighted 3 lines, 3 species, basically, 3 tips here. If I wanted all 3 of those to be part of the same clade, how far back would I have to go in history? Yeah, I have to go all the way back. I have to include everything that's up there, right? In order for those to be in the same clade, because that's their most recent common ancestor, and I'd have to include then everything that's descended from that ancestor, which is everything on the board. So that's the only way I could include those in the clade. Good, perfect. So most of you understood that pretty clearly. So this would be called polyphyletic. Means that it has multiple pathways that have led to it rather than just one. So this would also not be a clade because it doesn't include all of the descendants. So if it doesn't include all the descendants and it doesn't include the common ancestor, either of those would not be a clade. So these are things we worry about in science. All right, so I want to do a little activity in terms of trying to make your own evolutionary tree. So here are 3 organisms, 3 species. We have a bat, a seagull, and a walrus. Okay. I want you to think about how you might group them, how you might classify them. So if you were to draw an evolutionary tree of these, how would you group them up? Which means, which 2 do you think are more closely related than the other ones? Okay, so we have one option, which is bat and seagull together like this, and then you'd have your walrus like that. Any other ways you might group these? So what do you mean by that? Well, I don't mean how they're similar to each other. I mean, which 2 do you think are more similar to each other than the other one? All right, bat and seagull. Anyone have any other ideas? What do you think? Okay, so they're both near water, whereas the bat's on land. Sure, there's a different way. So how did you group these? Well, how did someone that wanted this, how did you group them? Okay, so these 2 have wings, right, and this one does not. So that's how they're. All right, yeah, but yeah, I don't want you to get into the. I'm asking for how you can, like, if you were to draw an evolutionary past of these, how would you group them together? So how are 2 of them more similar to each other than they are to the third? So one way of grouping them is saying that bats and seagulls have wings, so they're more similar to each other than they are to a walrus, which doesn't have wings. We have some other?
I think the bat and the walrus are kind of similar because the bat and the walrus the walrus doesn't really have wings, but their arms look more like the bat.
Okay. So you're saying that though the bat has wings, it kind of has arms, whereas a seagull doesn't really have arms as part of its wings. So the walrus and the bat might share some similarities that are actually different from the seagull. So you have the bat and the walrus together and the seagull separate. And you had the walrus and so seagull and walrus together, and then the bat separate just based on location. So two of them tend to be over the ocean, and one of them tends to be in the land. And that's a perfectly fine way of classifying things as well. So we have three different systems here. Anyone want to come up with anything else? Or? All right, that's pretty good. So we have three options, all based on different characteristics. So this is based on wings. This is based on kind of similar body parts. And this is based on location. Yeah.
I think the bat and the walrus too, because neither of them have feathers.
Okay, there we go. So feathers versus no feathers. And that'd be another way of doing that as well. So it's the same idea, but different reasoning. Right. Okay, so when I was thinking about this, I didn't come up with the land and sea one, which is a really good one, but I came up with this. And I actually was more on the lines of the feathers because walrus and bat actually both have hair. So whereas the seagull has feathers. So they do share that. So one is grouped by hair and one is grouped by wings. So in order to know which of these is the actual evolutionary past, and then by this, how should we name these things so that we can put them in groupings based on the right things? Because maybe we'd name them different based on which group we have them in. We might want to have bats and seagulls be in the same genus and have walrus be in a different genus. Or we might want to have walrus and bat be in the same genus and have seagulls be in a different genus based on which one that actually should fit into. So this is kind of the difference between these two things is one, an activity which we just did, which is a classification, which is we group species together based on shared characteristics. So presence or absence of wings, presence or absence of feathers or hair, location that we tend to find them in. Those are all shared characteristics. Phylogeny, this word, means making sure that the characters that we have chosen reflect the evolutionary relatedness and not just coincidence. Because we're going to learn as we go through this that things will share similar characteristics for other reasons besides their relatedness in terms of evolution. So they will have these similar traits. They might eat similar food. They might have similar body shapes due to some other reason besides that they share a common ancestor. In terms you might understand, this is the, what do they call them? Like brother from another mother or sister from another mister, not the way. So like when you have a friend that has so many similarities to you, even though you don't have similar, your families are completely different, right? But you have a lot of similar traits. So which one of these is wrong, or only one of these could be right? Only one of these did we pick out a characteristic that actually is representative of their evolutionary past. And that's what we strive for in this. So the characters that are not, the characters that are not from or due to evolutionary past are characters that are called analogous characters. They're results of something called convergent evolution and not relatedness. So bat wings and bird wings, although they're both wings, they are actually analogous characters. They are not due to a shared evolutionary past. And you're totally right in talking about the way the wings are shaped. The bat wings is actually a flap of skin between the arms and the body, or really between the arms and the legs. So it's a big flap of skin here. And that's how the bat wing is made. Whereas the bird wing is a modified arm, basically. Like it's a separate structure, and you'll notice it's got feathers on it. It's not skin. So the bat wing is made of skin, whereas the bird wing is feathers. So they're both wings, and they're both adaptations that allow for flight. but they have different evolutionary paths. So they are called analogous characters. Okay. So this one's actually not correct. Although it seems like the most logical one, right? That's why I picked these, is that it's not. It's just not right because they are different evolutionary lineages. So the opposite of analogous is homologous. And we talked a little bit about the word homologous. We used that before when we were in genetics. Homologous characters are characters that are the result of shared ancestry. So the wings of, it actually is an albatross, not a seagull. This is how good I am at identifying my birds. My wife would be proud. She studies birds. The wings of an albatross and a penguin are homologous characters, even though albatrosses use their wings to fly and penguins use their wings to swim. They have shared evolutionary paths. They both are from the same kind of evolutionary lineage. So they are homologous. So when we draw trees, we try to use homologous characters and not analogous characters to draw them, which is a very hard thing to do. Because oftentimes you don't know, like, is this character homology or not? And this is why there's been a lot of mistakes made in taxonomy and why people can be taxonomists and actually make a living out of figuring out which one of these relationships is, due to that, the right character, which one is actually tied to this. So when we make these trees based on homology, we can then actually put those classification names, the species, genus, family, and order, right onto the tree. So if we've done our trees correctly based on homologous characters, we can then say, this clade is a genus, and this larger clade represents a family. So here we've got coyotes and wolves, which are both in the genus Canis, and they'd be in the family Canidae, but they're in the order Carnivora. So everything that shares a common ancestor in Canis is in that genus. We have striped skunks, which are in the genus Mephitis, and we have European otters, which are in the genus Lutra, and they share a common ancestor that puts them in the family Mustelidae. And then we've got panthers up there that are in a different family than these other ones, but they're all in the order Carnivora. So as we go further and further into the past, we can say this clade, the clade that includes Canis latrans and Canis lupus, is one genus, Canis. The clade that includes Mephitis and Lutra is the family Mustelidae. And the clade that includes the Felidae, the Mustelidae, and the Canidae is in the order Carnivora. So those groupings, kingdom, phylum, class, order, family, genus, species, are all based on clades, and those clades should be all based on homologous characters, so sharing certain characters. Okay. So when taxonomists, we're going to go into this and talk about kind of how difficult this is. When taxonomists make these trees and name things, they try to reconstruct evolutionary history by incorporating as much data as possible. So that includes the shape of things, which is morphology. That includes DNA. That includes life histories. And they try to take it all into context because looking at just one characteristic can give them the wrong interpretation. But looking at several different interpretations, they can kind of settle on, well, more characters point to this relationship versus this relationship. But just like you guys did, and you came up with 3 different ideas for how these 3 species might be related, a phylogeny is a hypothesis. So these were 3 different hypotheses of how they might be related. And so with new data, phylogenies can change. And this has happened a lot recently. So we used to put especially in kind of the plant world. We now know, so algae, which I studied, used to be in its own grouping, its own kingdom, the Protista. But we now know that some algae are actually really closely related to land plants, whereas other protists are more closely related to fungi, and other protists are so different that they're in a different kingdom altogether. And so we've gone from having, at one point we only had like 4 kingdoms, and now, depending on who you talk to, we have 8 kingdoms or 10 kingdoms, and there's lots of different ways of splitting these things up. So the groupings and the namings of these things are constantly changing. Okay. All right. Okay. Just skip over that stuff and come back. Go on. So just to come back to this one again, we want our phylogenies should represent evolutionary history, and just like your family tree, phylogenies will be these branching trees where you'll be connected to other things by common ancestors. And just looking again at this, humans and cats here are mammals. Humans, cats, and amphibians are 4-legged vertebrates. Humans, cats, amphibians, and fishes are all vertebrates. And you can keep on classifying things higher and higher orders, and this should represent our evolutionary past, which is what this does. Okay. So, if you were to pick one of these 3 as primary purpose of constructing one of these evolutionary trees, which would it be? You probably won't pick 3. So what is my goal? What is the goal of a taxonomist when constructing a tree like this? Yeah, it's not just to group them based on functional traits that are similar to them, but it's really to determine the evolutionary history of a species or a group of species, and to really represent that. Because when we're studying organisms, if we know their relatedness, it kind of sorts out a lot of the similarities we have between organisms and allows us to really understand them to a greater extent. So we know more now about bats because we know that they're actually more closely related to a walrus than they are to a bird. Okay. All right, so that's really the major part of Chapter 14. Chapter 14, we talked about speciation. We talked a little bit about large-scale evolutionary pushes, including things like the movement of continents and what else did we talk about? Geological time and mass extinction events, things like that. And then I would like you to know just briefly a little bit about classification, because we'll get into that a little bit more. So that's really all I wanted you to know about Chapter 14. What I'm going to do kind of for the last little bit of today is tell you a little bit of a story about the field that I've done most of my research in over the last, I don't know, when did I start? 2000, so the last 14 years or so, which is the seaweed world. And use it as a way to talk about biodiversity in general, because this is what we're going to get into in the next few lectures. It's really amazing how little we know about our world in terms of biological diversity. So this is kind of the title of this section of it is why biologists have no idea how many species there are, and one way that we're going about changing that. And it's going to kind of focus on the stuff that I've been working on when I'm not teaching to some extent. So here's some pictures of seaweeds. If you were to group these together, how might you group them? Okay, you could group them by color. So which would you group together? We've got a red one, got a brown one, we got some yellowy brown ones, right, like that. You could also group them on, sure, we've got blades, kind of the two on the top are blades, and this one's very hard to see, but it's like single cells, and these are kind of tube-like, right? So you could group them like that, right? It's really hard in the seaweed world. Actually, if we look at genetics and we look at how these are actually classified, color is right to some extent, but these top two, one of which is kind of brownish, are considered red seaweeds. These bottom ones are brown seaweeds, and these two actually here, this one kind of microscopic one and this one that's actually a gigantic blade, are actually the same species. So they're just two different life stages of the same species. And red seaweeds and brown seaweeds are in different kingdoms. They're as different from each other as we are from them, or as we are from a fungus, something like that. Like it's that drastic how many differences there are between red seaweeds and brown seaweeds, even though that top one is brown and tubular, and so is one of the ones on the bottom, right? So in the seaweed world, it's very, very hard to tell things apart. Even for really highly trained specialists, something like this, which is what British Columbia looks like. So the western coast of Canada, this is what the beach looks like if you go there, is just these mats of seaweeds. This can be intimidating to actually know anything. I spent 5 years in graduate school working mostly in this environment, and I only know a fraction of the species that are out there by sight. And some of them I can only guess at because there's multiple things that look the same that are almost impossible to tell apart just by looking at them. And my supervisor, who's been doing that for 30 years or so, he still gets tripped up a lot when he goes into the field. So how many species do you think have been described in the world? How many species do you think are out there that we actually have names for? Think it's 50,000, 10,000? Do you think it's 10 million? It's somewhere in between? Would you imagine it's a lot or a little? You probably imagine there's a lot out there. And there have been a lot, but not nearly as many as there are. There's about 2 million things out there that have names. But estimates are that there's about 10 to 100 million species out there. That's a pretty big range, right? 10 to 100 million. So it's been 250 years since Linnaeus lived, and in total, we've described about 2 million species, but estimated number of species that are on the planet is anywhere between 10 and 100 million, and we really don't know. So we have no idea. It might be, I don't know, it might be a billion. We don't know. We know it's at least 2 million. We're pretty sure about that it's more than 2 million. Considering that when I was working up in Canada, 1 out of 5 things that we picked up was a new undescribed species, pretty good bet that we don't know very much. So we were constantly naming new things. Okay, so why do we know so little? Why do we know only 2 out of a potential 100 million? And why don't we know how many species there are? Well, one, it takes years of training to become a specialist in classification. So some very commonly studied things and things that people have a lot of hobbies doing, so like bird taxonomy is doing pretty well, though not as well as you might think. And like tree taxonomy does okay, but again, not as well as you might think. But most things on Earth have very few people doing it. So how many people do you think are out there doing seaweed work? Well, in Florida, let me see, there's one guy up in Tampa that's retired. There's me. Really, in terms of macroalgae, I think that's it. So there's a guy that sometimes does stuff, but he's more of an ecologist and not a taxonomist in Miami area, but that's it. So you think, okay, which is why, if you're going into research like this, pick something that hardly anyone does, and you're going to be the expert. So I've already gotten calls from people in the area asking me what stuff is. And a lot of it I still don't know yet because I haven't been here that long. But all of a sudden I'm the expert for Florida because I'm the only guy that's not retired that does taxonomy down here. So there's a very few people, and it takes years of training to become specialists. So even someone that's been trained in taxonomy, I go to a different place. I go from Canada down to here, and I hardly know anything that's around here. My wife's always remarking at when she picks up something and throws it at me and says, "What is this?" And I say, "Well, it could be this or it could be this." And I'm still not sure of myself. Each specialist can only identify a small fraction of species diversity, so there's just simply not enough people doing it. One thing is that there's no money in it. Like it's really hard to get paid to be a taxonomist. Usually you're teaching on the side, and then you do this kind of in your free time. If you're lucky, you might be at a research institution. You get to spend more of your time doing that, but there's not that many jobs for taxonomists out there. So very few people are doing it. And this is a picture of beetles. There's, let's see. There's about 350,000 described species of beetle. Just imagine one person. Do you think they'll be able to just actually identify 350,000 species by sight? No. Maybe 100, maybe 150, maybe 200, maybe 500 if they're really, really good and have a really good memory, but not nearly. So think of how many people's brains have to be focused on beetles to carry all that knowledge. It's really not something that happens very often. Another problem for taxonomy is that species are typically described by something called morphology, and morphology is the way things look. So the reason why this is a problem is that a lot of things out there have very simple morphologies, so they don't have much to them. So if you're describing an animal, you've got a lot of parts to look at. You've got just the structure of their skin, you've got their internal organs, you've got their brain, you've got where on their body their hairs, you've got all kinds of different things to look at. If you're describing a seaweed, it doesn't have eyes, it doesn't have a brain, it doesn't have feet, it doesn't have all those parts. It's very simple, and most things on Earth are pretty simple. A lot of things are single cells, so think of how hard that is. Conversion evolution, which is what we talked about here. So bats and seagulls having wings makes things that are different look similar. And then there's something called phenotypic plasticity. So remember, phenotype is how something looks. Things that are the same can look drastically different. I'm going to show you some examples of this as well. And also unknown life histories, and this is a big problem for seaweeds. Seaweeds can be, when they undergo meiosis, when they go from diploid to haploid, they're not just a single cell. So in humans, we're only sperm and egg very briefly in our lives, right? It's just prior to fertilization. In a seaweed, there can be entire free-living organisms that are haploid, and they can look nothing like the diploid organism, the other organism. So a lot of the stuff that I studied is a crust for part of its life, and it's a gigantic, like 20, 30-foot long bladed structure during the rest of its life. So we don't know their life histories. So here's just examples of simple morphologies. These are all different species. Very few characters to look at. These are just green circles, right? But they're all different species. And with very simple morphologies, it's very easy for two things to start looking the same. So convergent evolution. So here are 6 different species of red algae. Try to tell them apart. They're all red blades, right? And this is because being a red blade is a really good strategy for living. They can absorb a lot of light over a large surface area. They've got lots of reproductive tissue as part of them, but they're all different. They're all different species. Then there is phenotypic plasticity. So this is the ability of an organism to change its look in response to environments. So these two things are the same species, but this one is much more spindly than the one on the top. The one on the top is really bushy, thicker blades. It might live in a more sheltered environment, whereas the one on the bottom might be pounded by waves a lot, and so it starts to look different. So even though these are the same species, they look slightly different from each other, right? So, and then there's unknown life histories, which is what I was just talking about. We know very little about the life histories of some things. So this is an example of, on the top is the gametophyte, which is actually the diploid stage of this organism. And it is microscopic. So this is taken through a microscope. And on the bottom is the same species, and there's a scuba diver in that image. So these things are gigantic. These are these giant kelps called macrocystis, and they can be, 60 to 90 feet long. This is one plant. So they have different morphologies at different life stages. They can be microscopic at one point in its life, and it can be gigantic in the other part of its life. So these are very hard to see. Well, okay, even so, well, why do we care? Why do we care that we can't tell things apart? Because I'm just talking about seaweeds, and you could care less about seaweeds, right? You can tell most animals apart, and that's what you're really interested in, and you can tell a lot of trees apart, and they're kind of cool, so you can look at them. But you're never going to look at seaweeds, right? So who cares? Well, people that are doing things like making food might care, and people that are doing things like making medicines might care. So let's imagine you found a plant that has some unique property that cures a disease, and you go out and you collect. So you've studied this plant and you found out it cures a disease. So you go out to get more, and you find out it doesn't work this time. So you give up. But if you had really been able to identify those two, maybe those were actually two different species. And the first one has the curing properties, but the second one doesn't. And so you gave up on this line of research to this super duper cure because you thought, well, not every individual has this, so it's really something I can't harvest, right? So that's one problem. So unique compounds are found by finding these different species and describing them. So this thing called Taq polymerase, which is used in every molecular biology lab in the world, is a unique bacteria that lives in very unique environments, and we've been able to harness some of its activities to do molecular biology. There's also just biophilia, which is human connection to nature and other forms of life. We like to know what's out there, and we like to know how different it is, and it's fun to differentiate between them. And then, all right, so there's a lot of reasons why we should care about the biodiversity. Biodiversity, just preserving biodiversity, also has a lot of ecosystem services. Even though we might not know what's going on, doesn't mean these things aren't going on. So plants and different organisms will purify the air, will aid in decomposing waste materials. There's certain species to pollinate certain crops. So if we don't know what's out there, we don't know what species it is that is important for the crops that we're planting, how will we know if it's in danger? Or if we're doing something that's causing it to go extinct, or we're causing its population to crash. Different biodiversity is responsible for creating and maintaining soils, so there's lots of organisms that go into soil production and maintenance. And the reason why weather is not as extreme as it could be is because of a lot of biodiversity. So knowing what's out there might be important because we don't know exactly what everything does. And there's a possibility that we're headed towards another extinction event. So some scientists think we're headed towards a sixth mass extinction. So we talked about 5 major mass extinctions in the past. We've seen in the last 400 years over 1,000 species that we know of go extinct. Think of that we only know 2 million out of a potential 100 million. How many species have gone extinct that we never knew existed? So compared to the fossil record, this is about 100 to 1,000 times higher than during a non-extinction event. So there's pretty good evidence that we might be in a biodiversity crisis where many species are declining and very few are emerging. So, but we don't really know because we don't know what's out there, and we can't tell if what we're doing is impacting these species. So one thing that I've been part of and that is going on really worldwide now. It started in the United States and Canada, and it's now spread to most of the world, is something called our large-scale international efforts to catalog diversity. And one of these is the Census of Marine Life, and there's others as well. Census of Marine Life has been going on for about 10 years, and this was actually, sorry, I made this slide a few years ago, so it's about 15 years now. There's probably more scientists than this now, but there were over 2,000 scientists, over 80 nations. There's been, by 5 years ago, there have been 540 expositions, millions of dollars, and about 6,000 or more new species found over those 10 years. Pretty good chunk of organisms. The push that I was part of was called the International Barcode of Life, which is actually using new technologies to help us discover and identify new species, actually using DNA to identify new species, which really helps in the seaweed world and is much easier than doing it through morphology because it takes a lot of training to be able to identify things by sight. But if you can learn one technique, which is extracting DNA, sequencing DNA, and then use computers to tell the difference between things based on their DNA, this can really rapidly increase our ability to find out new species. And this started in 2003. So I got involved in this about 2005, so not long after it started, and it's been 4 years, so it's been almost 10 years that I've been part of it, but it's been about 11 years that this has been going on. So it gets its name Barcode of Life because it's actually seeking to barcode species like the Universal Product Code, like a barcode that catalogs commercial products. It's going to use a DNA sequence to catalog biological diversity. So a barcode in a grocery store, when you scan the barcode, it instantly tells you information about the product. It tells you the name. It tells you the price. It tells you how many are present in that store, right? It tells you a whole bunch of things about it. And a lot of you even have these things on your phone now. You can go around a store and you can scan a barcode and it tells you all about it. It tells you how many calories are in that product and how much weight you're going to gain when you eat it or something like that. So there's a lot of information you can get from a barcode. A DNA barcode, the same concept is being applied to organisms. So instead of a tag on it, it's going to be using the organism's own DNA. And by inputting this DNA sequence into a computer database, you'll be able to learn a whole bunch of information about the organism. So you'll be able to see pictures of the organism. You'll be able to see the distribution of the organism. You'll be able to find out if where you found that organism is rare or very common for it. You'll be able to tell how endangered that species is, how common they are, and all kinds of things about it. And you won't need a lot of specialized training. One of the kind of end seems pretty theoretical because I've been part of this for a while and I haven't seen any progress towards it, but what they originally in 2003 hoped to design is to have something like a phone, or like a handheld device that you could just take a sample from an organism, like pick a feather from a bird or take a piece of leaf off a plant, and you'd put it into the device and it would sequence the DNA and it would tell you all about it. So you would now not have to be a specialist to identify things. All you'd need to do is take a few cells from that organism, put it in your little handheld device, and it would tell you all about it. And that's kind of the end goal. And what it's using DNA. So DNA, which we didn't really go into, we kind of skipped over that chapter, but DNA is a sequence of nucleotides. It's a sequence of letters. There's 4 possible letters, and they're arranged in a unique way. And so by looking at a single region of DNA in all organisms, you can actually identify things based on a sequence of only about 650 bases, because as 2 species separate, their DNA separates. So species acquire unique mutations as they evolve, and so these 2 butterflies are going to have different sequences of letters, and then by looking at those sequences of letters, you can tell what they are. And actually, by telling how similar 2 species barcodes are, you can tell a lot about how closely related they are. So two things in the same genus are going to have different DNA sequences, but they're going to be pretty similar. Two things in different families are going to be a little bit more dissimilar. Two things in different orders are going to be even more dissimilar. Things like that. So this is what we do. Here's kind of a story from one of my colleagues, one of the graduate students I worked with, her research. Her pictures were prettier than mine, so I've used her stuff because red algae are a little bit prettier than the brown algae and the microscopic stuff I was looking at. But here's a family of red seaweeds called Calaminiaceae. And her research question as a graduate student was how many species are in Canada? And these are some examples of them. So our lab went out, and I went out with her as well to go collect. And we went and collected samples from all different areas. When we brought them back to the lab, we would press some of them so we could look at them and identify them based on traditional ways. But we'd also take little pieces of them, grind them up, and sequence the DNA from them. So we'd have a DNA sequence. We would also have kind of morphological sequences. We'd then put all this information, everything we knew about them, where we collected them, what they looked like, what their DNA sequence is, and we put it all into an online database. So it has a map with the location that we collected it, with what we identified it based on morphology, and we, as the experts in the field, were able to put the right taxonomy on it, and it would automatically compare our samples with sequences from anybody else that's collected those samples. So we now know where else in the world this species was collected at. And anybody that puts in a sequence that they don't know what it is, it'll tell you how closely it matches the species that are in this database. So one kind of implication for this is, let's say you're a border agent, and somebody comes in with a bag of green something or other. And you don't know what it is, right? You suspect they're up to no good and they're bringing in drugs into the country. So rather than making a mistake and arresting someone for just suspicion of bringing in something that's illegal, you take a sample of it, put it in your device, and it tells you, oh, this is oregano. So they just want to make pizza when they get where they're going. So they can move along very simply. So you don't have to be able to identify things, and you don't even have to have a whole organism. So a lot of spices would be very hard to identify when they're all ground up, right? But not if you have DNA sequences. So they could run it into this database and find out things about it. What we were able to do is able to build trees like this and find out when we had something that was never named before. So one of the genera that she looked at was something called Euthora. And you'll notice, actually, there's Pugetia and Calophyllus there. You'll notice that some of these have species names on them, but some of them have species names with numbers next to them. So we have Pugetia firma 1 and 2. We have Pugetia fragilissima, species 1 and species 2. We have Euthora, species 2. We have no idea what its genus was. Here's a Pugetia firma, species 3, that's very far away on the tree from the other ones. And we actually... we originally went into this in the early 2000s thinking, oh, we already know what everything is, so we're going to put it into this database and be able to identify it. And what we found was that we actually didn't know what anything was. So half the stuff, or even more, that we put in there was new species. And just by doing this genetic work, we were able to find new species and name them. This one, this Euthora species 2, I find this really hilarious. She named it Euthora timbertonii because she really likes Tim Burton, and she thought that it looked like something that he would draw. So there's a picture of it here. It's kind of this flower-like thing, and underwater it actually looks like it could have been in The Nightmare Before Christmas or something like that. It's very kind of creepy-looking flower. So we've been able to identify a lot of new things. So other applications that this technology has been being used for right now, in 2010, there was a study on the stomach contents of deep water sharks. So it used to be that if you wanted to find out what a shark ate, you had to go through its stomach contents, pull out all the little teeny bits of things, look at them under microscopes, and then really, really hope that you got a big enough piece that you could figure it out what it was. And you'd have to know a little bit about everything because, you know, who is an expert on every single fish species as well as every single invertebrate species that's down there? And you have to be able to separate things that were not organic from things that are organic. And these are all little teeny bits of things that have been digested by sharks. Now what we can do is we can just take those bits, sequence their DNA, run it through the database, and we can find out all kinds of stuff about the stomach contents of sharks. So basically, they found out that deep water sharks eat everything, from commercial fishing vessels, so bits that have fallen off of boats, to other sharks, to all kinds of other things as well that have just ended up on the ocean floor. There's also, we've been able to, let's see, 2009 there was a study on mosquitoes and looking at diseases that are found in mosquitoes and finding out which species of mosquitoes actually transmit diseases. So we didn't always know, and you could actually now catch a mosquito, suck out the gut contents from that mosquito, and identify diseases that it's carrying really easily, and identify other microorganisms that it's carrying pretty easily. So pretty amazing stuff that we can do now with DNA and this kind of idea of taxonomy. It's really opening up our eyes to how much biological diversity we don't know about and helping us to chip away at that 100 million species that we don't have names on. So just to kind of summarize, we're surrounded by biological diversity, but we're ignorant of most of it. We really need to improve our knowledge so that we can use biodiversity appropriately. So there's lots of things out there that we don't know could be useful to humans. And also we want to conserve it because we don't know what's going to be useful down the line, and we want to make sure we're not destroying areas that might be important. And these new technologies like the DNA barcode and large initiatives will help us do that. And I want to end with a little funny thing on kind of the consequences of misidentification and why being able to properly identify things is pretty important. So here's a clip from a show that you might recognize. Let me see if this plays well. Okay.
What's this single-celled creature you got here?
Ah, the glowing substance Sid found on Emma Pierce's foot. It turns out it's bioluminescent dinoflagellates, a luminescent algae.
Found in marine water.
Uh-huh.
All right, so this is a CSI clip, and they identified this bioluminescent dinoflagellate that is a marine organism. So they knew exactly where the kill occurred, and they were able to catch the perpetrator. One problem, that wasn't a bioluminescent dinoflagellate. The thing on the left was what was on that screen. It's a desmid. The thing on the right is a bioluminescent dinoflagellate, which looks nothing like a desmid. Yes, bioluminescent dinoflagellates can be found in marine waters, but desmids are only ever found in freshwater. So by misidentifying what was on the corpse's foot, they've now completely arrested the wrong person, right? So misidentification can be really, really bad. So even though these things are small and hard to identify, they can be important for crime scene investigators. All right, so that's the end of my talk today. So next class, we'll actually start looking at some of the biodiversity. We're going to start looking at bacteria on Thursday.